Voila. Yeah, it looks like it's doing recording. Yeah, we're all set. Here we go. So the peritrich ciliates that we're going to look at, what I want to do is I want to basically talk a little bit about the ectoparasites of snails. And I'm going to also talk a little bit about the endoparasites of snails today. So one of the things that we can do with these ectos of snails is we can I don't understand why I can't get this my screen to go full screen with this peritrich ciliate thing. Sebastian, do you know?
Let me check it.
Can you see it there?
Not full screen. Not full, but well, almost. I think
Almost.
You can really.
You can?
Yeah.
Oh, okay. So this is a paper online by Bianca Sartini, and there's a couple of others that have been written by some of my friends down in Argentina. This one is Brazilian, but there's also one that was recently published in Argentina by one of my friends who's the curator of the collection in La Plata, Argentina. And she published a paper on peritrich ciliates of some of the species of Physa from Argentina. And this paper is interesting because it was published more recently on the ciliates of Physa in Brazil. So it's kind of interesting. They're almost pretty close to the same kinds of papers. The nice thing is that they are published fairly close together, and so that means that they are. It says my stream has reduced audio quality. So the papers were published fairly close in time together, so that means they're comparable at this point. So that's kind of cool. So I'm going to see if I can get my screen to work. Here we go. Do this. I don't know why it has to do it twice. Can you see that? Is that up?
Paper, yeah.
And it's up now. It's moving too.
No.
It's not?
No.
Okay, so I have to go back here and
I see your mouse.
there we go. Now we should have it.
Scroll down.
Here we go. There, that should do it. Yes. But why can't I operate? Not sure why I can't operate my screen. Anyway, so let me get to the part of the page that has so you can't see it at all now? I'm moving the screen.
Now it's working.
It is? Jeez,
Yeah.
I don't understand it. Okay. So basically the objective of this paper is a summary about, and it's talking about epibiosis. And that's a word that we don't ever use. So we call them ectoparasites or ectosymbionts. And this is one that's brand new, and they're calling it epibiosis, and they're making a new word here, basically. A facultative association widespread in aquatic environments involve epibionts, that organism that remain attached to the surface of living substrates. I suppose it's reasonable, but I call them ectosymbionts instead. And a guy named Wall, 1989, published that. So, and this is basically during the sessile phase of the life cycle, because a lot of these protistans, these colonial flagellates, or colonial flagellates, are have a stage of their life cycle where they're sessile, and even though they're sessile, that means that they don't move. The host moves around, so they're able to move all over the place because the host is moving. It's fairly interesting. So I guess these are ciliates, epibiotic ciliates. So let's move down. The reason I'm showing you this is because these are on Physa, and I know that some people had a hard time killing Physa, but if you look at this, this is pretty funny. This is the funny thing here, control plus. Okay, what do you see that's interesting about this? Snail sampling. What do you see? What this first sentence there what is the weird part of that about that first sentence? That you can get a whole paper on one sample. Well, the funny thing is they only collected 140 phyces. And that's a really low number. That is what some of the students here collected in one day a couple years ago, last year. They were doing 250 specimens every couple of
Yeah,
hours. Those guys
Karen.
were smashing FISA like you couldn't believe, and they got thousands of individuals for their statistical tests. So 140 FISA is what they published this whole paper on. So what I'm saying is, if you guys take really good data, then you could publish a paper. You know, especially if you send this one journal. So you have to be careful about what you send your papers to. All right, so let's zoom back out. Does that make sense? Can everybody hear me? Anybody?
Yeah, we can hear you.
Nobody can hear me?
Yep. Yeah. And the student answered there.
I guess I'm not hooked up to my. Well, if somebody says something, then pull it up because I don't hear anybody. So let's see if I can fix that. I don't know why that is. Headphones. Test. Nothing. Test. I have one set up working and the other one.
No, we couldn't hear you.
Very strange. Okay.
No, we can't.
Now you can hear me?
Yep.
Couldn't hear me before? You were hearing me through your speaker, maybe. Is it okay now?
Yep.
Yeah?
Now it's working.
Okay. All right, so here we go. Okay, so they did a spatial distribution analysis, which there's some interesting things here for our in our case. The snails were individually placed in petri dishes and observed for the presence of epibionts using a stereo microscope. So another thing you don't want to say, and when we're working, is if you put something under a microscope, it's very difficult to see it because it's under the microscope. The microscope is sitting on top of the specimen, smashing it. So you don't ever put anything under a microscope. You put it on the stage of the microscope, but you don't put it under a microscope because it smashes the anak out of the microscope, or out of the specimen that you would put there. So that's another thing that, you know, if you're writing science, you need to write science. Don't write like you're in 4th grade. Okay, here is what I wanted to show, and this is cool. So there is a FISA. It looks just like our FISAs we know and love from around here. Everybody agree?
Lighter, maybe.
Yeah. So a little bit darker, but then here's the conical part, and you can see these specimens on there, and you can see when you zoom way in on them, you can see how pretty they are. Then you move in even further, you can see them. Nice, pretty pictures that they used here, and there's some Bordetella, I think is G, that's Bordetella, and then there's some others. We'll get some names here in a second, but these are, let's see what their scale bars. So these are 150 micrometers, 1,500 micrometers. So there's some different sizes of these animals here. So D through L is 25 micrometers. So right here, the length of this, this is 25 micrometers right there. So that's, oops. So this is basically 25 micrometers from here to here. That's pretty small. But we can see those easily with our scope, with our scopes we have. And so these things are peritrich ciliates, they call them. Epistylus is the one that I keep forgetting. That's a really, really common one. Epistylus, and that's D here. Epistylus comes in these clumps. Has anybody seen these yet? No? Anybody say anything? It seems like I'm still muted or something. Somebody say something. Okay. I can hear you too. I was just wondering whether you could hear me or I could hear you. So anyway, Epistylus, D, what's E? E is opercularia, another one that should be easy to remember. Apparently it has, the cool thing, it has an operculum. right here. So this little thing pops off, and out comes its little cilia to then bring food into its inner chamber. Here's another one you can see. So these are called opercularia, and F is what? F is carapace, and G is our vorticella that we know and love. We can find all of these in the aquatic environments in which you guys will be looking, if you look careful enough. But as you notice, this vorticella, G here, is only, G is, B, C, D, that's 25, yes, this is 25 micrometers right there. So that's 25 micrometers. So that's going to be on 10x, your 10x lens on those little microscopes. This will be pretty good size. So you'll be able to see these pretty easily. So here's I. I is Epistylus plicatus, and J is another one, Thericola. That's a beautiful one. It has it has like a tunic here, a little external body covering, and inside comes these things. And the thing that's weird about these is these are single-celled organisms. These are not multicellular animals, but they look multicellular. But when you look at them, all they have are organs. I'm sorry, all they have are organelles and one big cell or parts and pieces of the cells. They're not multicellular, which is amazing. So they do have colonial parts. So this part is one cell. This part's another cell. Maybe I can do this without doing that. This part's a cell. This part's a cell. This part's a cell. So they're colonial cells. They do stick together. And they may share food through this stem part of their body. So anyway, I think that we're going to find these. Last year we found lots of them. And I think you can find them we're going to find them this year. So this is, the reason I wanted to show you this too is because here we go. Look at this, the table right here. Prevalence, intensity, mean abundance, discrepancy index. Now, I don't know what a discrepancy index is. I've never heard of that before. So we're going to have to go back and look at the materials and methods to figure out discrepancy index. We can ask our resident statistician and see what he thinks. Our resident statistician says he has no idea, never heard of a discrepancy index before. I mean, we've done a lot of statistics. I've had years of statistics. I've never heard of that term. So let's look at it and find out what it is. So let's find discrepancy index. So I think I can do by Control F. I'll just look up index because I don't know how to spell discrepancy. To access epibiotic community structure, that's a bunch of using nouns as adjectives, we analyze the data on prevalence, mean intensity, mean abundance, and the discrepancy index. Poulin, say, all becomes clear. For each epibiont species with the quantitative parasitology 3.0 software. That's great. So we should download quantitative parasitology 3.0 software. In all analyses, colonial peritrix were quantified considering each zooid as an individual. So there we go. They didn't define what a discrepancy index is at all. We have no idea what it is. We just know that it occurs in a computerized package, but we don't know what it means at all. Very strange, but there you go. But the neat thing about this paper is that we have these pictures of these things, and we can find these same things in our snails. So, all right, so that's what I wanted to talk about there. Now, another thing that I wanted to discuss with you guys today is the definition of species. And many people call species, well, many researchers have different definitions for species. So some things we define species as a group of interbreeding organisms that have their own evolutionary tendencies and their own evolutionary fate. Some others define species as phylogenetically cohesive groups of individual populations. And so it's interesting because there are about 40 commonly used species definitions out there. the species definition we use is mostly one that's called the biological species definition, in which the species, individuals of the species can reproduce with other individuals of the same species. And that's kind of the one that we use mostly. But the one that we use when we do our evolutionary studies is a phylogenetic species. And that means that these groups of individuals are derived from a common ancestor. I'm going to make, do you have that going?
Yeah, we're ready.
Okay, so they're derived from a common ancestor. Do I need to change it, or can you make it go live?
I will go live in my stream. From my
Okay.
Can you see the whiteboard?
No, I'm standing in front of it.
Can you see the whiteboard? Let's see on my Discord account. You get it?
Okay, so I'm not going to put definitions down. I'm going to draw pictures. Oh, I wasn't saying anything. Can you hear me now? Hola?
Yeah.
Yeah, so can you hear me now? Yeah, I just was silent. I was kind of enjoying the moment. Kind of, just kidding, actually. You know, basically, so um, can you see this phylogenetic tree?
Yeah.
Okay, so here, let's go, we'll put these things, B, C, D, E. So these represent, this group represents species number 2, and this group represents species number 1. And so in this case, there's characters that define this species. So we could pop on characters right here that would define these species. They could be DNA characters. They could be behavioral. So some of the characters that you can use to define species would be molecular. It could be behavioral. They could be morphological. Molecular, behavioral, morphological. So those are the primary ways that we could define species. Not many people use behavioral traits to define species. But a lot of people use molecular traits because they're too lazy to work to get the molecular traits or morphological traits. Morphology, usually phenotype can be thought of as equaling phenotype equals genotype um, times environment. So the genotype times the environment interaction makes the phenotype of the organism. And so if we can define what the genotype is and we know what the environmental interactions are, then we know what the phenotype is. The phenotype is morphology, what it looks like. Phenotype means what it looks like. Phenom means what something looks like. So phenotype equals genotype times environment. So the environmental interaction then influences heavily what the animal looks like. Sometimes the environmental interactions, the environmental part of this equation can be minimal, can be low. Sometimes it can be very high. And that's how we then get morphological variation due to environmental variation through time and space. So when we're doing our work, we look at all three of these molecular characters. And if we look at molecular characters, aren't molecular characters simply little tiny morphological characters? What are molecular characters? Well, A, T, C, G. Well, when we really look at those, adenine looks like, we have a sugar hooked on with double bonds with another sugar, and they're all hooked together. When we look at them carefully, we see that they're tiny morphological structures. They're tiny chemical structures. So basically, when we look at DNA, molecular characters, we're just looking at little teeny tiny morphological characters at the same time. Because these things hook up together and they don't hook any other way. When we do PCR, for instance, we break the DNA chains apart, we multiply up the DNA, we hook it back together, we get more DNA of the same kind. So anyway, my point is that molecular, behavioral, and morphological characters are all equal, can all be used in equal weight to determine phylogenetic relationships among organisms. And once we have good data on phylogenetic relationships among organisms, then we can start saying something about our biology. So in this case, these species, A, B, C, D, E, are related in this way. There was an ancestral species that gave rise to A plus B, C, D, and E. And then here, there was an ancestral species here that gave rise to K, L, M, and N, all equal. And so there was one that separated here and one that separated here. So there's different ways that species form. And the hypotheses of species formation are legion. There's lots of different hypotheses about how species can actually form. So modes of speciation is what I'm talking about. And so the things that we talk about when I teach my systematics class are speciation, different kinds of speciation, allopatric speciation, centrifugal isolate speciation, peripatric speciation, sympatric speciation. I mean, we could just keep going on and on. But right now, what we think most of the kinds of speciation that occurs in the world as we know it, we think most speciation that's occurring, the model or the mechanism of speciation, we think is allopatric speciation. Allo means different, patric means space. Allopatric speciation means speciation in different spaces. So if we start off with a population like this, bunch of individuals out there running around, mating or not mating, eating, not eating, being preyed upon, not being preyed upon. And then suddenly, over a 2,000-year period, a river runs through it. Isn't there a movie? A river runs through it. A river runs through it, separating those populations. So now we have two populations separated by a river. And it turns out that these things don't swim. So they're non-swimming individuals that are in populations. We don't know what they are because we haven't studied them well yet. But those little X's just don't have enough gumption to be able to get out there and swim. So we have this population being separated in space and time. So if we were drawing this, we would have time 1, and then this is time 2. So time 1 was here, where everybody was happily interbreeding in a large panmictic population. Everybody should know that word. Who knows the word panmictic? Has anybody heard of the word panmictic before? Who's heard of it before? Really? Not true. Jared? You've heard of it? Did you know what it meant? You've never heard of it. Okay, good. Well, and you are a senior? Yes. So how could you guys get through 4 years of biology at University of Nebraska and never heard of panmictic populations? What was that? Well, no, I'm not talking about that. I'm talking about panmixis at this point. Yeah, well, panmixis means the potential for any of these little X's in this population to mate with another potential X in the population. So, for instance, humans are considered a panmictic population. We have the potential for mating with any other person, any other individual in the any other sex individual in the population of the world. So we're considered panmictic. There are some isolated demes where people don't go very often, like up in the Himalayas, where it's very, very isolated because of geographic isolation. But most people are called panmictic. In this case, panmixis has been stopped. And we stopped this, and now we have two populations, a bunch of populations of X's here, and a bunch of individuals of, these are individuals of X in two different populations. But these are X1s now, X2s. So X1 and X2. So there was a disjunction in the population here where the panmictic population is then completely separated in space and time by a river. The river came through in about 2,000 years, and about 20 million years later, there's a big canyon there stopping those little X's and the little X1s and the little X2s from making it across. So they can't make it across anymore. So after about 2 million to 20 million years, they had separated into different species. So that's called allopatric speciation, in which the species are isolated in space and we have new species forming because of the isolation mechanism. Now, humans are kind of messing all that up in the world because we keep moving mussels around, we keep moving mice around, we move insects around, and so that we're, instead of having this allopatry, we're making big sympatric groups of stuff now. So it's difficult. But most of the animals and plants out there that we see have pretty good allopatric isolation when speciation happens. So this is basically how species form. And if you haven't had this class, you should take my systematics class, which I'm going to teach next in the fall of 2021. So you can log in because we'll do it live. So, okay, so we have these different species that form by allopatric isolation here, a bunch of them formed by allopatric isolation there. We have all these characters that define these species. If we used a phylogenetic analysis program, we could analyze the matrix and we'd show these relationships. What are the characters called that define one whole clade on a tree? Anybody remember that? What that was called? You should have learned that in like high school. The character is called a synapomorphy. A character that defines a species is called what? Don't everybody speak simultaneously. It's called an autapomorphy. So there's synapomorphies and there's autapomorphies. So an autapomorphy is a character that defines a species. The autapomorphy for humans is our ability to reason. That's really the deepest one for us. But there are others also, chromosomes. There are also genetic phylogenetic, or there are also genetic characters that only humans have. So an autapomorphy for humans is our ability to think and reason, and that's a behavioral characteristic. Morphological characteristics of humans, there are others too. I mean, we can separate ourselves out from chimpanzees pretty easy by the length of our arms, the length of our tibia and fibula, etc. So, but autapomorphies define the species. Synapomorphies define groups of species. So in a synapomorphy for the primates would be, well, how about a synapomorphy, a little bit easier. A synapomorphy for the mammals would be what? Hair, fur. And the synapomorphy for the birds would be flight, right? Would synapomorphy for the birds be flight? No, because what else flies? Flying fish. No, not, yes, feathers. There you go. Synapomorphy for the birds are the feathers. Synapomorphy for the mammals is hair or fur. Birds don't have fur. Mammals don't have feathers. Birds sometimes have teeth, though, so that's not a good one because some of the beginning birds had teeth, so we can find those in the fossil record. So those are a couple of the synapomorphy. There's a whole bunch more we could talk about. So speciation, what is a species? So a species is, in our definition, we can kind of use two definitions, two parts of the definition of species integrated together. One is a phylogenetically separate group of individuals that are reproducing and have their own evolutionary fate and their own evolutionary tendencies. And so there's no crossing like this. Nope. No crossing like that. Nope. So there's no crossing out of those unless, of course, sometimes there's unreasonable crossing when people put things in zoos and they can't help it. But in natural systems, the behavioral systems are set up to stop that. Molecular systems also stop species crossing, and sometimes morphology. So that would be something like, if it was a morphological character, it would be post or pre-mating isolating mechanism, for instance. Okay, so a species is a group of organisms with their own evolutionary tendencies, their own evolutionary fate, and they're a phylogenetically cohesive group that are held together by morphology, behavior, and molecules, which basically informs everything else. The biological species is these things can actually reproduce together. So we can integrate those two together to have an operational definition of species. Okay, so that's what I wanted to talk about species. And this, what I did just now, was about a 2-hour lecture from my systematics class down to about 15 minutes. So let's go on. Any questions about species, speciation, synapomorphies, autapomorphies, that kind of stuff? Plesiomorphies, symplesiomorphies, all those things. Everybody's already had all this, right? Someone tell me if you haven't had this, if you really haven't worked in phylogenetic systematics at all. If we work hard. So that's our idea. That's why we're doing this. Okay, so I need to jump and change this. Okay. So I need to go to video. See what my video looks like.
Click Epson there.
Yeah. Change windows. And I'm going to go to the other screen.
It is on there.
Which one?
On your screen. Now we have the...
We have this?
Yep.
Oh, let's see it. Huh, that's interesting. I like that. So it should come up here now. There we go. So I'm going to go here, and so now you can make a choice. So, well, what we're going to do is we're going to just look at these different kinds of cercaria. So remember that these, well, let's see, do we have a good life cycle of one here? Oh yeah, there's one. There's a good life cycle. I can move this one vertically so you can see it too. So this is a good one. Now, I've just lost where we were on the other one. That's okay though. We're going to zoom in now. Zoomeroonie. I can go like this. Move it over here. Oh yeah, this is a cool machine. All right, so in this case, we have a kingfisher. And it doesn't look like it's in focus to me, or is it just my... Okay, we have a kingfisher. Everybody knows what a kingfisher is? Maybe you can't hear me? Nobody can hear me. Oh yeah. Okay. Yeah. So a kingfisher is is a bird that eats. Okay. What order is the kingfisher in? You didn't have. I thought you had 121. No. A kingfisher is in the order Carassiformes. They are in their own order and they have unique structures. These things are amazingly diverse throughout the world, and they're all in their own order, which is interesting, Carassiformes. So I took ornithology when I was a freshman in college, and I got an A in the course because I was so interested in the class. But I didn't realize that you weren't supposed to take like graduate level courses when you were a freshman. Nobody told me. So um, anyway, so this kingfisher poops out a ciliated egg, and out of the ciliated egg comes, this is backwards, it's interesting. Out of the ciliated egg comes a, what? Remember it starts with an M? Somebody say it first, you'll get 2 points for the day. Oh no, Augie, you can't say it. Augie wants the 2 points. Miracidium. A ciliated miracidium comes out of the egg and then penetrates the snail. And then it goes into the snail and turns into a sporocyst. And then it turns into a cercaria. And then it turns into a free-living cercaria. You see the little tail on it? This is the kind of cercaria that if you went swimming in one of these ponds around here, one of our students did that a couple years ago. She jumped in the pond. She was one of my honor, well, she wasn't an honor student, but we published a couple of papers together. She jumped in the pond and got massive amount of swimmer's itch because she got infected with all these cercaria that penetrated her skin. So she wasn't happy for a while. Anyway, so then the cercaria penetrates the fish and goes into the muscle. And this is a really common one called uvillifer. Is that backwards to you?
No, it's on the your computer.
Why is it backwards to me? Huh. Weird. Okay, anyway, uvillifer is the name of this trematode, and it has a life history of going from the bird to the snail, to the fish, to the bird. So that's how this life cycle goes. And when we're out here walking around, every one of these birds, every one of these carassiform birds, kingfishers here, is infected. And if you go out to some of these ponds here, and if you know your fish, who knows what kind of fish this is? Who's a fisher person? You guys all sleeping? Come on, you have to take a guess. Good guess. I think that it's a bass, but usually crappie are a little bit more narrow, but it could be a crappie, but I think it might be a smallmouth bass. But I'm not a bass fisherman. I usually used to hunt or fish for salmon in the Pacific Northwest. So, but anyway, good guess. You got 5 points for your guess. I'm gonna write that down. 5 points. So I would throw out M&Ms if you were here, but there's no M&Ms, so I'll just eat them myself. Okay. So anyway, this bass gets eaten by the kingfisher, and there we go. But they have to be baby bass because they get into a big bass. Can a little tiny kingfisher eat a big bass? No. So, okay, there we go. So that's an example of a life cycle. And where would we be able to, if we were interested in stopping this life cycle, for instance, because there's a lot of people in the world who get schistosomiasis, which is caused by a trematode that lives in people's bladders and also in their large intestines. And there's about 400 million people right now infected with that in the Middle East and South America. And so where do you think the most likely place to stop that life cycle would be? Well, probably the easiest place would be to stop the pee and poo from going into the water to go back into the snails. I think that's the easiest way to increase the hygienic quality of the area where people live. That's probably the easiest way. one way is to kill all the snails. So that means just like dump something in the water and kill them all. That'll end it right away. So some people really had a great idea. They took crawdads, crayfish from North America, and threw them into Africa, and the crayfish go into the ponds and eat everything in the pond. That works pretty well because it wipes out the crayfish or wipes out the snails, but it also kills all the biodiversity in the pond. So that sounds like not a very good idea, but they're doing it now, believe it or not. Anyway, so what else were we supposed to talk about? Oh yes, I want to talk about what these different things look like. So this is a really crazy looking sporos, redia or sporocyst inside this snail for this uvillifer. But I'm going to show you some of these others now, if I can find it again. Near the clam. Past the clam? There we are. So, let's see, let's go up here. So when you guys are smashing your snails, you'll be finding all these things. And these animals have, let's switch to the board. So can you switch to the board for everybody?
Yep, we are still in the camera. We need just moving it.
Okay, just move this camera.
Yep. I can do it.
Okay, so here is a trematode. This is called a trematode. Trema means what? Who's taking Latin? Trema. Good guess. Who said it? Hi, Kelsey. You get 5 points. But it was wrong. But still, you said something. So if everybody speaks up, they get all kinds of points. Look, we could have points rolling in like mad, or catch some M&Ms. Close. That plat means platy is flat. 5 points for you. Was that Jared or who, John or Felix? Who was it? Ah, Felix. Yes, I got you on the last one. Okay, so it doesn't mean flat. So platy equals flat. Platy helminthes. Platy means flat. Helminth means worm, flatworm. So trema means hole. So trematode means animal with a hole. So this has 2 holes, actually. These are called digenetic trematodes. That means trematodes that have 2 animals in their life cycle with holes. So that's what it means. But this is a trematode, and this is the ventral sucker. So Felix, you got like a pack of M&Ms too, okay? So what I should do is, keep track of this stuff, and then at the end of the semester, everybody's going to get a box of M&Ms in the mail. All right, I could really do that. In fact, I might. So remind me, Sebastian. Okay, ventral sucker, and this is called, it's near the mouth, so it would be called the what? What do you do? Which kind of hygienist do you go see to get your teeth cleaned? An anal hygienist? No. You go to a what? What kind of hygienist? Oral hygienist. I tried to give you a contrast, not an anal hygienist, but an oral hygienist. Okay, so oral hygienist. Oral sucker. Okay, so after a couple of weeks of taking a class like this with me, you guys would all get it. But unfortunately, we only have a few days, so I have to go really fast. Okay, all trematodes have an oral sucker and a ventral sucker, which is pretty interesting. And when you look at some of these Cercaria, you see these little suckers, and I'll show you that. Well, you actually can see it on the screen there if you flip over to that one screen. Yeah, you can see them. These little suckers, there's one here. Look at those. There's one there. Oral sucker, ventral sucker, oral sucker, ventral sucker. Yes, that's good. Nice. So you can see the oral sucker and the ventral sucker right there, and a stylet even. So inside of this trematode, there are also other things like other kinds of things like ovaries, vitelline gland, um, Malus gland. So, and this is the cecum. Does anybody know the Latin root of cecum? Cecum means blind sac. So, cecum is a blind sac. So, like well, our cecum on us is we have a cecum where the small intestine goes into the large intestine. There's a little lump there called the cecum. Then off of the lump there is the appendix. So the appendix is hanging off of our cecum. And the cecum is just this little blind sac. Sometimes we get an inflammation of the appendix and it's pretty bad, but lots of times that's where the pinworms hang out. They like to be in that part of the intestine. So anyway, trematodes have two suckers. They have cecum. They have a double cecum. They only have a single entrance to their digestive system. So it's two-way out of the same hole. So they feed, and then they have to regurgitate out the same hole the waste products from their digestion, which is kind of disgusting. So they don't have an anus. So flatworms don't have an anus for the most part. There's maybe one or two that have been shown to have an ani. That's plural for anuses. So, okay, so that's what these characters are here. We see an oral sucker, we see a ventral sucker, and we see this thing here, which is the cecum. And here it is again. The cecum is branched. It goes down to both sides of the animal. And so some of them have a very hardly branched cecum at all, and some of them have an extremely branched cecum like this guy. You can see how the cecum branches all the way down. This is an echinostome. So what's echino mean? It means spine. What's stoma mean? It means mouth. So echinostome cercaria means cercaria with spines around the mouth. Okay? So echinostome cercaria are those cercaria with spines around the mouth. And I guarantee that if you look enough and you're collecting in a place where there are muskrats, you will find echinostome cercaria in the snails where you're doing your work. So that's kind of an interesting thing just there. So this is just a standard flatworm. I'm going to erase it because it's not a very nice looking one. So I'm going to get rid of it. But Trematoda. So it's gone. So that's so anyway. So you'll see lots of these. You may see lots of these different kinds of cercaria. These guys have little stylets, and you can see that with your little microscope that you have. You'll be able to see the stylet. These are called Plagiorhynchus types of cercaria, Plagiorchiid types of cercaria, and they have these little stylets in them that help them penetrate their host. Then there's these others that have these split tails. Those are called Furcocircus cercaria. And the cercaria that has a single tail, a cercaria that has a single tail like this one, is called Ziphidiocercus cercaria. And this one is called a rat tail cercaria. That's called a rat tail cercaria. What's the name? Oh, it's called Rattenkönig cercaria, or rat king cercaria. Some cercaria don't even have a tail at all. Some of them have this Furcocircus type, which is the split. Some have this Ziphidiocercus type. Let me zoom out. Okay, the Phytiocercus. Some of them have these little spines on the cercaria, a little bit of a tail. So these things that are echinostome cercaria are easy to see because they have this little bit of a banner on the end of the tail, plus they have spines around the oral sucker. And this is, anybody have a guess what those little dots are there? And remember, these things swim around outside and they need to go up to the top of the water column to find their host. How do they find up? Well, in this case, they use those eye spots to go towards the light. So these are eye spots, primitive eye spots in these animals. So that, in this case, these animals have little eye spots in it that allow them to orient themselves in the water column by where the sun is. Pretty cool. When I was just starting at the University of Nebraska, a guy called from Fremont Lakes. Anybody know where Fremont Lakes is? Up near Fremont, Nebraska. There's a bunch of lakes there. And he said, go ahead. Oh, wow, you know where right there where the lakes are, just north of the river. Well, geez, I forgot his name. I was thinking about him earlier. Well, he called me about 15 years ago and said, I'm having a problem up here. Can you guys help? And I said, sure. So I jumped in the car with my graduate students. We drove up there, and he said, what, my daughter was out here with her kids, and they were in the canoe, and they had their hands in the water, and they went in. The next day, their arms were completely broken out with pustules all over their arms, but only on their arms where it was into the water. And I said, I know immediately what your problem is. He said, what is it? I said, you have cercaria in the water that's being released by the snails that are in your water. So the life cycle is duck, snail, duck, snail. So it was obvious what the problem was because I looked over there and there were about 50 Muscovian tame and white ducks, Peking ducks, swimming around, being happily ducky, they're doing their duck thing out there in the water. And I said, there's two ways that you can get rid of this problem in your water. He said, what? I said, well, you can poison the water and kill all the snails and most everything else in it, or you can get rid of the ducks. So he called me the next spring and said, I said, how's your water? He said, all the ducks are gone and we haven't had the problem since. So thank you very much, Dr. Gardner. So it was a really gratifying experience to be able to just go up and just solve the problem. The reason they had so many ducks is they weren't using duck birth control. So the ducks kept reproducing. There were more and more and more of these tame ducks around there, and the ducks didn't fly because they were like, flightless ducks, the white kind, And so they just kept getting more and more ducks, and they were producing more and more poop, and the snails were becoming more and more infected. So every snail we looked at was infected with the cercaria of these echinostomes, and also some of these other, not echinostomes, but the schistosome types of cercaria, the ones that occur in the duck guts. So it was pretty interesting to be able to solve that problem right there. So just a little bit of knowledge can go a long ways to helping people out. So if anybody's ever gone into some of the ponds around Nebraska that are warm in the summertime and stay up near the top, you get all these pustules on you. Now you know where they come from. They come from, well, trematodes. So these guys penetrating your skin. Yeah, ask me. Uh-huh. Yeah. I go like this. Yeah, that's it. Nothing you can do. You just have to wait until your body finishes its immune response on those things. And hopefully you're not immunocompromised, so you'll have these things going into you and living in you for a while. So if you're immunocompromised and you get some of these things, they can actually cause a really lot of problem. But usually your immune system will handle them right in your skin, and all you'll have is some skin inflammation. What you can do is you can take cortisone or put on cortisone or take prednisone, which is a heavy-duty corticosteroid, which stops inflammation, and so then you don't have as much inflammation going on. So a couple different things you can do, but those are over the counter. You have to talk to the doc about that. So, but you can use some ointment, which is what, 1.5% cortisone cream, or maybe 0.5% cortisone cream. You can't really get very high level cortisone cream over the counter, but you can get some. So that's it. That's about all you can do, unfortunately. So also, you don't want to be snorting mud in a warm lake. And why would that be? Well, because of Naegleria fowleri. Everybody knows that, right? So, okay, what else? If anybody likes to know about Naegleria, let me know. So I think that we'll stop, because what I wanted to do was give you a good introduction to those trematodes that you're going to be finding in your snails, and we're going to put a bunch of those pictures of the trematodes up. I think we already have up onto Canvas, so you can look at these different pictures of trematodes that are going to be in the snails. So there's Echinostome cercaria, and there's Zophidiocercus cercaria, and there's Furcocercus cercaria, and there's Schistosome cercaria, and there's all these different kinds of cercaria that are out there. So anyway, any other questions? Sorry about your family getting those cercaria in their skin. I feel bad about that. But as I said, all these white marks on my skin are from ticks. If ticks went extinct, I probably wouldn't be too sad. Yes, that was a beautiful picture of a nymphal tick. It was wonderful. Did you kill it first, or was it like wiggling on there? Yeah, yeah, sorry about that. Yes, yeah. So the best way to identify a larval tick is use a lighter and then burn it. I hate larval ticks. That's what all these marks are. So, yeah, from larval ticks from Bolivia. I walked next to a stick that had about 10,000 larval ticks on it, and I didn't know it, and they just streamed up my arm, taking little bites of lymph, then fell off, and I didn't know that I was going to be miserable for over a week. I mean, not just, go ahead. Yes, because what they expect is to get on a small rodent, take a bite, and then fall off, and then they metamorphose and turn into a nymph, and then they do the same thing. So, yeah, larval forms just take one bite and then go. And they don't even take blood, they just take lymph. And the lymph part of your skin is the most immunoreactive part of mammalian skin. It's intense. You probably had dogs before with mites in their ears or intense itching that they've had larval ticks or chiggers on them. So, yeah, no, I wouldn't count them, but what you could do is you could do some qualitative estimates, or you could even do some quantitative estimates by estimating what you have on a piece of stuff, go through and do it for a period of time. Use some thought on it. Use some interesting, think about ways that you can do it on your own to come up with a qualitative estimate. Like, don't go in that don't go in that field because there's 10 million ticks per square inch. But we can go to that one, there's only 5 ticks per square inch. Basically, just kind of give yourself some way to make a quantitative estimate that then you can use for like a qualitative screaming match and run away from them. I hate ticks. So how do you keep them from getting on you? You're using like DEET like mad? Massive. Yeah, so you're going to have to put it on long pants, long shirt, DEET, 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 on gloves, everything. Because we had them, yeah. Yeah, I would make some estimates and find out what you have. You're not going to be able to figure out the species probably very easily, but just go through and since you've already done it, you have a bunch of data. You could say, 1 minute of dragging produced this many ticks on 1 meter of cloth. You have some good data right there, and you can then make some estimates and amplify it up and see what you've got. So I would just play with it. Get your well, 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 who knows? I mean, you don't know. The thing is, I'm thinking time, I'm thinking area, I'm thinking just all that stuff. So, and also, I'm thinking I'm really itchy right now. Just thinking about it. So, yeah. Yeah, stick the duct tape. Yeah, and see like 2.5, 1.5 inches of duct tape, 4 inches wide on that thing. Pull it up and then estimate what you've got. Do that. Make some rough estimates, and that would be interesting. See what other people have done with that in the literature. That's what the way to go. See what else has been done. Check the literature first. Yeah, you could look up density of larval ticks in, where are you? Arkansas? In Missouri, density of larval ticks in southern Missouri or whatever part of Missouri you're in, um, or measuring density of larval ticks using, or measuring density of larval ticks. Just go into Web of Science and see what you can find. You could go into Journal of Entomology. They sometimes publish things on ticks. Just and go into Google Scholar, see what you can find. I bet you'll get thousands of references because people work on ticks a lot because ticks are really important in human and animal welfare. So important. Absolutely. Yeah, do it. But don't throw away your data. You have good data, keep it and play with it. Yeah. Yeah. Sure, yeah, I wouldn't. Don't spend too much time on it. What I would do next is I would go out there with about a 55-gallon barrel of gasoline, spread it across the field and light it and then run. Ah, too bad. I know, I'm just kidding. But ticks, yeah, because you're going to find them on your skin, no matter what you do. Even though you put on all that DEET and everything, you're still going to get them. So get in there and take a shower and get them off you. You're going to have to do a tick check with some trusted people because they're going to get in places you can't see. So uh, yes, but you might get a big pustule like me. I don't know. So all right. Well, thanks for that. I'm really itchy now. So I think I'd rather have a tapeworm in my gut than a larval tick stuck in my skin. We should have like a poll. Who would rather have a tapeworm than a larval tick? Well, tapeworm just sits there in the intestine and absorbs stuff. It doesn't really do very much. But a larval tick, on the other hand, is sucking your blood. I don't know. So, or lymph, I guess if it's a larval form. So any other questions from anyone? Now you all know that there's lots of different variation in trematode morphology, larvae, the cercaria. We know there's lots of variation in how they reproduce. We know about species. We know about speciation. We know what a species is now. We understand how species form. Basically, we've had a whole systematic biology class in 1 hour. So, okay. Well, if somebody wants to do a phylogeny of something while they're working out here, that'd be kind of fun to do too. Like, what are the phylogenetic relationships of some of these trematodes? You could make a big matrix based on all their characters and analyze it using mesquite and then with TNT. That'd be kind of a cool thing to do. So let me know. Yeah, go ahead. Yeah, yeah. I want everybody to read it, and you could just submit one sentence. Everybody to submit one sentence of the Brooks paper from somewhere in the paper that you've said, "Oh, wow, that's interesting." Is that the one where it's finding them before they find us? Well, I'm on that paper, so everybody should read it anyway. Well, tell me what you think. DAMA, DAMA. That's the protocol. So, anyway, here's my little babies. Can you guys see this? Is it working?
Yeah.
So these are little cute mosquitoes that I started growing from eggs down in Lincoln. So I put them in here on the 10th of July as eggs, and now they're almost ready to pupate. I feel so proud. I haven't identified them yet, so that's the next thing. Maybe tomorrow, maybe tonight, we'll try to figure out what these are. So, am I missing anything? Anybody? This is your chance. You've got me up against the whiteboard. Ask the questions. What is the meaning of everything? What is the meaning of life and everything? Does anybody know the answer? 42 is the answer. Have you guys read Hitchhiker's Guide to the Galaxy? Well, if, there you go. You should have said 40. Yes, it's 42. 42 is the answer. All right, so thanks for being here, everybody. I wish you were right here, but you're here. So we appreciate it tons, and I'm going to give it all I can. We're giving it all we can to make this worthwhile. Thanks for being here, and we'll post this later if my recording worked okay. Take it easy. We'll see you guys on Monday, and ask questions over the weekend. We're going to go up to Crescent Lake this weekend and try to look for larval acanthocephalans that live in amphipods. So that's what we're going to go for this weekend. All right, everybody. Hasta pronto. Muchas gracias. "42.
42.
Yeah, it's warm in here. It's nice. I like it. Feels like we're in the dojo.